In this video, I am going to tell you briefly about what I did for my PhD thesis. I isolated and purified the ribosomal RNA gene from the sea urchin sperm. And that sea urchin was called Litocynus variegatus, Atlantic sea urchin. You know that this is a marine animal. Sea urchins are available in Florida and Bermuda Islands, among other places. So we got them from these two areas. The sperms were collected in gallons and they were stored in liquid nitrogen tank and taken for isolation and purification of the gene. The first task was to isolate and purify the bulk gene, that is the entire nuclear gene, the gene contained in the nucleus of the sperm. Once that was done, then the second task was to get that particular gene that I am interested in, that is the ribosomal RNA coding gene from the bulk DNA. It was already established that this class of gene is resident in the heavy satellite DNA. What is heavy satellite DNA? When you spin the entire DNA in the cesium chloride solution for about 96 hours or so at a high speed, you find that a small segment of the DNA gets separated from the bulk DNA and occupies a different position in the centrifuge, that is in the heavier side of the centrifuge, which means that this particular satellite has a higher buoyant density and which corresponds with the higher guanosyne cytosine content. So that satellite DNA was fractionated, isolated, fractionated, and of course further purified by subjecting it to repeated centrifugation. When analytical purity was established, then the next point was to see whether this gene indeed codes for ribosomal RNA or whether any other gene surreptitiously or adventitiously came in that satellite band. That was tested with, of course, hybridization experiment, and it was indeed found out that the gene that was isolated and purified in the test tube actually belonged to the ribosomal RNA gene. In other words, this particular DNA codes only for the three classes of ribosomal RNA, that is 16S, 28S, and 5.8S. After getting that gene in, of course, gene copies in tandem sequence, because you know these genes occur in multiple copies, 150 copies, 200 copies, and the genes get further amplified at the time of oogenesis. And you can see 10,000, 15,000, 1 million copies of the same gene. But in sperm, of course, there are only about 150 to 170 copies. I was supposed to get a sufficiently long piece of DNA containing 4-5 genes randomly arranged. I did that, and then after that I did this hyperchromic spectral analysis. It is a sophisticated analysis. It was first developed by Felsenfeld and his workers in Maryland. We had the papers available. Based on that, we also followed the similar technique, and the data that I obtained, a huge number of data, the data were fed in the computer. Of course, for this I had to learn computer science and I had to develop the program in PL1 language. I did that and fed the data in computer, and the results that I got allowed me to draw a graph. And while making that graph, I realized that it has got an inflection at one point, indicating that there is a segment of the gene which contains very high GC content, and that high GC content does not really match with the GC content of the transcribed part. So this particular gene must have a segment which is not transcribed. This was the idea. But this was further established by examining these genes, tandem genes, in the electron microscope. The DNA was selectively denatured. Of course, denatured, it means that it denatured at 45 degree, 55 degree, 65 degree, 75 degree, and 78 degree, 80 degree, 85 degree, and so on and so forth, to see which area of the gene is denatured. And of course, denaturation is a temperature dependent process. At a very high temperature, you will see the GC regions breaking up, forming blisters. So the mapping was done. It was really a tedious exercise, time taking, but then the melting profile was obtained, a denaturation map was created. That map also showed that there is a portion of the DNA which contains very high GC content, and this if you measure the entire length of the gene, including that one, the region that contains very high GC content, you will find that the gene is about 7.2 million Daltons, whereas the real transcribed portion is of only 5.2 million Daltons, around 5 million Daltons. So what is that extra 2.3 million Dalton part doing? That part of the gene was described as NTS, non-transcribed spacer. And that was my finding. This finding startled others because the predecessors had not found this region. I was the one to find it out, and that became somewhat startling to others. But I had the data, a huge amount of data, and I did these experiments repeatedly and had come to this irrevocable conclusion. I put my ideas before the thesis committee members. They asked me to go ahead and write the thesis on it, but when it came to the time to defend, that is when I was asked to defend my thesis. All these examiners, 5 of them, came and pounded on me and asked all kinds of questions, and I answered them very patiently and very convincingly, very nicely, and I finally prevailed over them. And they finally realized that what Nirmal is saying is okay. So they finally said, fine. They asked me to sit outside the conference room, and after 5 minutes, came out Mike Blyman with a piece of paper and my thesis, loosely bound thesis, and I said, Nirmal, now you submit the thesis to the University of North Carolina office. And in that paper that he handed over to me, I had the signatures of all the present examiners, including himself, and the final remark was that the thesis is acceptable for the award of PhD thesis. So that was the, you can say, you shouldn't say shock of the life, but of course, it was tidings from the heaven. I didn't know whether to feel happy or to feel sad about it, because after being pounded so heavily, a few minutes ago comes the news that the thesis is acceptable for the award of PhD thesis. So it was a great experience, no doubt most memorable. Next day, I submitted my thesis along with the report to the university office, and then, of course, I waited for about 2 months to get my degree, because the convocation was going to be held pretty soon. That was okay. But then, after submission of the thesis, I also wrote a manuscript based on my findings and submitted it to the Journal of Molecular Biology, Cambridge, hoping that there would be some kind of response from the referee, or maybe my paper gets published, and if it gets published, that will be a wonderful thing in the world. After my thesis, after a few months, I returned to India and got busy with a number of activities. After 6, 7, or 8 months, when I didn't hear anything, I wrote to the editor of the Journal of Molecular Biology that I had sent a manuscript. What happened to that? He said, well, the manuscript has been sent back to the University of North Carolina. Then I wrote, why? I had given you a different address, India address, so why did you send it? But then, of course, there was no point arguing. It was done and it was done. Then I I was resigned. I was a little bit depressed, but then I recollected all those things, and of course gathered my energy and rewrote the manuscript and submitted to another American journal. They rejected it. The referee's remarks was, of course, sort of superfluous, you can say, or extremely critical. From the remarks, from the wordings, I realized that somebody does not want this work to be published. My sixth sense told me that. I modified the thesis according to the remarks, of course, but then I sent it to another journal. That journal also rejected, giving some funny remarks. And my hunch was actually asking me that somebody is really playing the role of a villain somewhere, but it was difficult to say anything against them. In the meantime, I received a letter from one of the referees. It was a private letter. It said, listen, your work is okay, but get it published elsewhere and be quick about it because it's getting late. He was hinting at something. I didn't quite understand. So I rewrote the manuscript. I sent it to still another journal, this time Experiencia in Europe, Switzerland. The paper got published, and that came to me as a huge relief. Then I published other papers also. After some time, I came to know that in my laboratory in the University of North Carolina, some people have been employed to do the work which I had done to find out whether indeed there is a spacer in the gene. And 4 or 5 research scholars worked very diligently, very hard, and ultimately, and they published a number of papers, and they ultimately corroborated my findings. But the irony of the whole thing is that they did not mention my name. Morality ethics says that if somebody has worked earlier than you make a reference. You can say that it was found out by someone else, however tentative the conclusion may be, but then the reference should be made. There is no reference at all of this work being done earlier in the same lab by someone else, something like that. It was shocking to me. And the most shocking of all was that the credit of finding out or seeing the non-transcribed spacer for the first time and that gene was given to a lady research scholar who is now a distinguished scientist in USA. Through ResearchGate, I found out the lady, her email address, and I wrote to her. I said, listen, you worked in the same lab, and of course, you must have seen my data and my results and everything, and the advisor must have told you something about my work. How is that my work, which was published, of course, not published in 1973, but the thesis was written in 1973, and thesis is supposed to be a published work because the data are preserved in Michigan. So how is that you became oblivious of all that? And that the same finding has been already seen in 1973 and in 77, 78 later years. You say that it was done by you, and you are the first one to do it. No, is it okay? And she started giving, please listen, I was working there, and it was not possible for me to counter them, and I was pummeled by them, and they dominated. And she said other things also, that being a lady, she was a bit harassed and so on and so forth. I don't want to go into that, but then she said, finally excused it. Let us forget what happened. Let's forget about it. I said, okay, my work also stands published somewhere else. And the whole world knows whose work has shown non-transcribed spacer for the first time and who has been given credit for that. But how is that the entire group wanted to crush this kind of news, or suppress it and give credit to someone else and not give any reference to my work? This is something very strange. Cannot be digested very easily in scientific world.